Cellular energy is a complex topic in biology and requires extensive and detailed examination. In a simpler sense, it would be easier to imagine how cellular energy functions when commenting on the two primary types of energy, that being kinetic energy and potential energy. Kinetic energy is energy when something is in motion, and potential energy is when something has the potential to use its energy as kinetic energy, such as standing onto a cliff, but not actively falling. A certain type of potential energy that cells use to complete tasks is chemical energy, which is the energy found in stored bonds of a compound. These bonds enable the cell to complete tasks and store energy for further use later, such as pumping a heart. First, let us start with a broad view of what energy is and how it is used. In this image, there is a general diagram of how we receive energy. Energy from the sun is brought up onto the earth through the rays of the sunlight, although some of it is lost as heat. The producers, or the plants, use photosynthesis in order to make glucose and to provide stored energy for later use. The consumers, or the animals, are those that consume either the plants or the plants consuming their own byproduct of glucose. And then the decomposers consist of fungi, bacteria, and worms. And everything else, after a certain amount of time, is lost as heat. Yet, of course, nothing comes without its price, and this is made apparent when consulting the laws of thermodynamics. Each time energy is used, heat is released as entropy, or the measure of disorder in a system. This energy is unusable and can no longer be used as any form of energy. Fun fact, a theory of the death of the universe is when there is no longer any energy or any usable energy in the world and everything is now heat, or entropy, hence the entropy death of the universe. Anyways, let's refer back to the study of energy and energy transfer involving physical matter. The first law states that there has always been and always will be the same amount of energy in the universe, and energy cannot be created nor destroyed. It can be transformed from one type of energy to another, but energy can never be created. The second law of thermodynamics explains that, as stated before, energy transfer is never truly efficient. There will always be some sort of loss of via heat or entropy as unusable energy. One main way energy is contained is via adenosine triphosphate, or ATP, which is an extremely unstable compound from which phosphate is released. And in correlation, a lot of energy is released via the chemical energy stored in the ADP, which was converted into ATP after the phosphate group was added. ATP is the primary source of energy for all cells and enables them to complete all their tasks. When talking about cellular metabolism, it is necessary to speak about the infamous enzymes and their empirical jobs that aid the cell by using its energy efficiently. On this image, there lies a general diagram of an enzyme being activated by a substrate linking up to its active site. It later begins a catabolic reaction by breaking the bonds and leaving behind the products, or the byproducts. What we are seeing here is an enzyme aiding in the catabolic reaction of a substrate by lowering the required energy to break these bonds. By adding to the active site and using this induced fit of the enzyme, the substrate with ease breaks apart. Rather than breaking these bonds without energy, as is thought from common belief, enzymes slowly lower the activation energy necessary for this reaction to occur. Picture this. The sun rays are raining upon the sky. There, a leaf captures the sunlight and photosynthesis begins. A lot of energy is transferred from the sunlight, yet some of it is still lost to heat. But then the leaf starts to do something called photosynthesis. Then in later respiration, organisms break down the glucose to use up as energy via ADP to ATP. ATP is a highly unstable compound that when breaking these bonds to use this energy, it is called a catabolic reaction, while fusing these bonds are known as an anabolic reaction. Generally, photosynthesis is known as consisting for anabolic reactions, from which it consumes 6 CO2 molecules and 6 water molecules, and with the power of sunlight produces sugar in the form of glucose and 6 oxygen molecules. This is all done with 2 different reactions, those being the light-dependent reactions and the light-independent reactions, or in other words, the Calvin cycle. The light-dependent reactions occur in the thylakoid membrane, while the Calvin cycle occurs in the stroma of the chloroplast. See, the chloroplast contains of many different structures that enable photosynthesis, those being the inner membrane, the outer membrane, the granum or a stack of thylakoids, the thylakoids, the stroma, the inner membrane space, and the lumen, the stuff contained inside of the thylakoids. And then with these cellular structures inside of the chloroplast, we can create photosynthesis. The light-dependent reactions have a series of chemical reactions that capture light via photosystem 2 and photosystem 1, in order. Photosystem 2 captures the ions from the sun, and it completes a catabolic reaction that separates water into oxygen and hydrogen. With the permeable membrane, the hydrogen is kept inside the thylakoid, while the oxygen is left free to leave, which inevitably creates an ion gradient. With this gradient, there lies ATP synthase, an enzyme that with an ion gradient creates ATP from ADP and phosphate groups. While this is happening, the electrons used to separate H2O are recharged at the photosystem 1 and are used to create NADPH from NAD plus and hydrogen ion. These products are later used in the Calvin cycle, or the light-independent reactions. In this reaction, the products of the light-dependent reactions are used to produce 1G3P, which when at a sufficient quantity can form glucose. The Calvin cycle utilizes the ATP and the NADPH produced earlier, and when it is considered as the most inefficient compound to ever exist, RUBP, to create glucose, which when used by cellular respiration releases a lot of ATP. All cellular life we know of partakes in what we know as cellular respiration. The series of reactions that bring us the chemical energy needed to complete these tasks and function properly. Through cellular respiration, scientists have estimated on average of 36 ATP molecules per glucose molecule. That is, if everything were to work at an efficient pace, which it never does. When focusing on cellular respiration, it revolves around these 3 major series of events: glycolysis, which can be aerobic, meaning the using of oxygen, and anaerobic, meaning a cell is not using oxygen, the Krebs cycle, and oxidative phosphorylation, or in other words, electron transport chain. With this formula in front of us, it is clear that we will be discussing the aerobic side of the cellular respiration, or cellular respiration that we as humans often use, at least when we're not running a marathon, from which we produce lactic acid instead. Glycolysis involves the use of both glucose molecule and 2 ATP molecules to break down glucose into 2 pyruvate molecules, and is present in the cytosol of the cell, while the other reactions occur in the mitochondria. After the series of reactions, the pyruvate 2 NADH molecules, not NADPH, and an extra 4 ATP are produced in the reaction with a net gain of 2 ATP, and they are brought to the Krebs cycle. The Krebs cycle is an aerobic process that requires 2 pyruvate molecules and oxygen, and with those transforms energy into usable molecules for later use. This is done via oxidation of the pyruvate, which is generally just adding oxygen to the pyruvate, and the use of multiple enzymes to have the byproduct of 2 CO2, 4 ATP, 6 NADH, 2 FADH2s, and citric acid, hence the name of the Krebs cycle / citric acid cycle. Finally, in the transport chain, the cells use the NADH and the FADH2 produced from the earlier reactions to power the electron transport chain, that with a permeable membrane creates an ion gradient for a total of 34 electrons if used at maximum efficiency. The electrons from these electron carriers are used to power the protein channels to gather up the ion gradient, hydrogen ions, which with ATP synthase produces glorious amounts of ATP. And that is cellular energy in a nutshell.